The double helix is a pair of parallel helixes intertwined about a common axis. Like right here, there's one, and connected to is another one. And DNA nitrogenous bases consist of adenine, thymine, cytosine, guanine, and for RNA it's uracil. DNA backbone. The backbone of DNA is made up of deoxyribose, which is sugar molecules connected to each other using phosphate, like these, and they're connected through covalent bonds.
And DNA nucleotides are binded together through a hydrogen bond because hydrogen bonds are weaker than the covalent bonds that bind the nucleotides to the rest of the chain. And there are two types of the nucleotides. There's purines and pyrimidines. Purines can be identified because they have two rings, and pyrimidines can be identified by their single ring. And purines consist of guanine and adenine, and pyrimidines are cytosine, thymine, and inside RNA it would be uracil.
Okay, and then there's a process called DNA replication, which the DNA copies itself for cell division, and then it starts through an enzyme called topoisomerase, which straightens out the backbone of DNA, as you can see here. And then helicase is another enzyme which pulls it apart, and then the single-strand binding proteins keeps it apart. And then RNA primers are put on it temporarily, and then taken off with DNA polymerase 1 or replaced with DNA nucleotides. And then DNA polymerase 3 just adds more nucleotides to the leading strand.
When the DNA is separated, it separates into two different portions: the leading strand and the lagging strand. The leading strand, the DNA polymerase 3 adds nucleotides continuously, going from 3' to 5' based on the placement of the sugars. On the lagging strand, it starts with RNA primase, which adds short sections of RNA, and in order to allow the nucleotides to be added through DNA polymerase. And then it's removed by DNA polymerase 1 and replaced with, which adds new nucleotides back, and then the DNA ligase holds it together, acting as the glue, and then exonuclease proofreads and makes sure there are no mistakes.